exercise it sounds, but 3 separate exercises in the lab manual. So the origin of animals, sponges, and some other the basal animal chapter. So look through all of those. Okay. So there we go. We can go with what lights we have on. There's not too many fancy color photos or anything this time around. All right. So last time we talked a little bit about about animal reproduction, sexual versus asexual, monoecious versus dioecious, all of that. How sperm is transferred through either direct mating or is broadcast spawning or through spermatophores. Now, once the mating has taken place, then we have the next sort of stage in an animal life cycle, which is its development or its ontogeny. All animals are, by definition, multicellular, and most of them, except for sponges and placozoans, have specialized tissue, and mesozoans we don't have to really work with. We'll get to those eventually. And most also have even more complex organ-level organization, although that's not in cnidarians or ctenophores. That means that you go from one unspecialized ovum, a fertilized egg cell, a zygote, into a complex multicellular organism. And that process is known as development, or the ontogeny of the organism. And we're interested in development, one, because it's a pretty complex, important process for animals. We want to know how you get from A to B by itself. But development can also help us understand how multicellularity might have evolved, how those complex structures came to be. And at least traditionally, although it's turning out not to be perhaps as useful, we've also looked at differences in development as an indicator of the potential relationships among major animal groups. And there we go. All right. So when we start with the egg, one thing to keep in mind, one thing that's important about eggs, the technical term being ova, ovum singular, ova plural, is that eggs are not a perfectly uniform cell, but they actually have a orientation. That is, they have 2 ends. So one end is called the vegetal pole, not vegetable, but vegetal pole. And that is the end that generally has somewhat higher yolk density, and is typically associated with the formation of the digestive tract, as we'll kind of see. And the opposite end is called the animal pole. It generally has lower yolk density and is where most of the other embryonic structures basically develop from. And so there's a sort of an orientation. There's an animal pole and a vegetal pole in a typical egg. And that helps orient the processes of development. All right, so the first thing that, of course, happens to that early zygote is that it begins to divide, the cells begin to divide, and so you have cell cleavage, the division of the cell. There are 2 kinds of cleavage, holoblastic versus meroblastic. Holoblastic means you cleave through the whole thing, that is, the entire zygote divides. The division passes through the entire egg, like is shown here. The top one is holoblastic cleavage. In contrast, meroblastic cleavage is where the divisions don't actually pass through all the way. And this occurs when there's a fairly strong, big difference in the sort of yolk content or nutrient content between the animal pole and the vegetal pole. In cells with holoblastic cleavage, again, the vegetal pole might be a little bit denser, but it's not substantially different from the animal pole. But in lots of eggs, there may be a very, very thick of a chicken egg. There may be a very dense layer of heavy yolk at one end of the vegetal pole compared to the animal pole. And the cleavage generally just can't penetrate through that. So all of the cell divisions basically take place at the top, and the yolk remains relatively undivided. So in a chicken embryo, for example, you have that huge yolk that's undivided. There's no cell division through it. And all of the cell divisions of the embryo take place on the top of that ovum. Okay. So, and as a general rule, so these diagrams are shown in the same size. Probably as a general rule, eggs that are relatively yolky like this tend to also be larger because you have your basic cytoplasm plus then all of the nutrients stuck below it. All right. So beyond beyond that division, we can also look at whether those initial cleavages. So the first two cleavages are always going to be from the animal on the animal-vegetal axis. That is, when the first cleavage is always going to go from the animal pole to the vegetal pole, or divide it lengthwise, essentially. So if you think of the animal and vegetal poles as the ends of the egg, then the first two cleavages, this one and then the one that's perpendicular to that first one, always run along the animal-vegetal axis, and those tend to be relatively equal sized. And then the third division is going to be horizontal, basically, and separate the animal from the vegetal pole. So first division, second division, third division. That third division in particular may be relatively unequal and produce different sized daughter cells called blastomeres, or it may be relatively equal and produce similar sized cells. So again, that's another variability in the very early stage of development. So here we're at the 8-stage state here. This is after 3 cell divisions. All right. The other big other thing to consider in that first set of 3 cell divisions is whether the cleavage is radial or spiral. So so we have a radial on the left, spiral on the right. The first cell division and the second cell division basically look the same in both. That is, you have one cell division to divide the original cell into 2 daughter cells, a second division that divides each of those, so you have a total of 4 daughter cells. And then the big difference here comes at the 8 cell stage. In radial cleavage, the transverse division is basically strictly horizontal here, and the cells stack up on top of one another from the animal to the vegetable pole here. And so those cells form a nice line. In spiral cleavage, on the other hand, this cleavage here is actually at an angle. So when this cell divides, it doesn't divide right here, it basically divides right here and produces one cell that's offset from the other one. So in the lateral view, it would look like this. If you're looking at it from the polar view, looking down from the animal pole, for example, the cells basically seem to switch over and sit in the grooves between the cells below them. And again, they might be the same size or different size, and that's the case. It can be the case in either one of these. Okay, so this is radial cleavage. The cells are lined up, and spiral cleavage, the cells basically are offset. And then in subsequent, so the next 2 cell divisions are going to again be are vertical, and then in the cell division after that, you will again have a spiral cleavage, but it will be counterclockwise as opposed to clockwise. So it will basically be in the opposite direction. So they alternate clockwise and counterclockwise as they divide. Okay. As it turns out, generally cells, organisms that have spiral cleavage also have what we call determinant cleavage. In determinant cleavage, each cell's fate is basically fixed by the time of the first cell division. So that first cell division divides into right and left halves, and the left half of that cell will only develop into the left half or the anterior or dorsal or ventral or whatever it turns out to be part of the organism, while the right half, the other half becomes the other half of the organism. In indeterminate cleavage, which is almost always associated with, not sure there may be exceptions, but generally associated with radial cleavage, up to the 8 cell stage, each cell basically has the potential to develop into a full organism. So, with that in mind, do humans have spiral or radial cleavage? You've got enough information here to figure it out.
Spiral.
Spiral, how do you know? Do we have what?
Determinant cleavage?
Do we?
Yes.
Which kind of cleavage can form identical twins?
I guess it would be indeterminate.
Indeterminate, right? Because If you split if the cell space is fixed by the first division, if you split those two early cells, one's only going to produce half the organism, the other's going to produce the other half. In indeterminate cleavage, you split those cells, they can still develop into a full organism, potentially even up to 8, although by that time you might have, volume issues of not so much cytoplasm, but, so since humans can produce, the only way to produce identical twins is to split after the first cell division, to split those cells apart, and each develop into a full embryo. And so that means that humans have indeterminate cleavage, because we produce identical twins, and that means we have regular cleavage. Yeah.
Is that where we have like stem cells that can develop into different things? Is it not?
Well, technically, I guess an embryo zygote is going to be a stem cell because it's going to develop into everything. And even with determinate cleavage, you still have, those cells can develop into any kind of cell, but they have some chemical cues that say only produce the front end of the body or the right side of the body or something. So they only produce half the organism. They just don't have, they've already kind of differentiated which part of the organism they want to be, but within that part they can still develop. None of these are specialized cells. So stem cells come later on, once the cells start to differentiate. Stem cells are those that haven't differentiated yet into particular cell types and could potentially develop into any kind of cell type. All right. So as these divisions kind of accumulate, you have more and more cells, and usually, and again, there's going to be a lot of sort of usuallys and most of the time, normally and typically in this kind of talk here. The next stage you reach is called the blastula stage. And at that point, usually the dividing cells form a hollow sphere with the fluid-filled cavity on the inside called the blastocele. Cele is just a general term for cavity here. So blastocele is the cavity inside a blastula stage. It's usually hollow. It's usually about roughly the same size as the original ovum. Maybe a little bit larger, and that's because that ovum has used a lot of energy to kind of divide up into smaller and smaller cells and to kind of develop, and so it's often not that much. This is not that much, even though it's many more cells. Each cell gets successively smaller as it continues to divide. Sometimes there are variations. It can be the blastula can sometimes be a more or less solid mass of cells inside, so that's found occasionally. For those cells, for those eggs that are heavily yolky, like a bird's egg, for example, most of the blastula stage will just have this sort of dividing cell sitting on the top of a massive undivided yolk. These kinds of variations are not as common. We're not going to worry about them beyond just to point out that there are lots of variations on this basic theme. But we'll go with mostly the typical blastocele, which is a hollow sphere of cells. That's kind of the next stage here. All right. After you have that hollow sphere of cells, your next process is called gastrulation. And gastrulation is the process by which you basically form those first two embryonic tissue layers, the endoderm and the ectoderm. Again, typically, notice that word here, there are exceptions to this, especially if you have like a solid mass of cells, or you're sitting on top of a yolk, then you're going to have gastrulation that works a little bit differently. But the typical sort of gastrulation, where you start with that hollow blastula, is that you have an invagination. Now, some of the cells at one end kind of push inwards. So imagine taking a balloon and sticking your finger into it, not so that you pop it, but so that it kind of indents. And so you form this indentation here. Cells push into the blastocele and take up some of that space. And so as you do so, you form, first of all, two layers. You have the outer layer, which becomes the ectoderm, and you have the inner layer which becomes the, really, I don't know why this book, where I stole these from, so it uses ento instead of endo. Endo, I think, is more common, but you have the inner layer here that forms the endoderm, and you have an opening here, and in the cavity that opens to the outside world, but inside type cavity here called the archenteron. That archenteron is basically the ancestral gut. That's what becomes a digestive cavity. So you have the gut, you have the endoderm that surrounds the gut, and you have the outer ectoderm. And then the rest of that fluid-filled cavity is the remains of the blastocele. So now we're starting to form some of the basic features we might find in an organism. The endoderm and ectoderm, the basic sort of germ layers or tissue layers, the basic gut here, and an opening, a mouth, or you might say. All right. So so in diploblastic layers, basically that's kind of in diploblastic species. That's kind of where you stop. So, this here is not all that different from a highly schematic view of something like a jellyfish. That is, you have your gut here, you have your mouth, your tentacles that hang down here around the mouth, and so on. And inside here, you would have a mesenchyme, which is a gelatinous, mostly acellular material that can be somewhat jelly-like, can be harder or softer, depending on the kind of thinner or thicker, depending on the kind of cnidarian you have. But in diploblastic species with 2 cell layers, that's all you have. And that intermediate layer is mostly acellular. It can contain cells or extensions of cells such as fibers and so on, muscle fibers. But those cells and fibers that it does contain are derived from the ectoderm. They come from the outer layer and extend inwards into that mesenchyme. In species that are triploblastic, that is, they have a 3rd layer, that 3rd layer is then what's called the true mesoderm, and the cells that make up that layer are derived not from the ectoderm, but from the endoderm. So they're moving in from the endoderm into that. And there's a couple of different ways in which mesoderm forms. We'll get to it here in a second. In addition, as a mesoderm forms, it can also form body cavities as well. So the way that the seal of the body cavities form tends to be tied fairly closely to how the mesoderm itself forms. So we'll look at those together. All right. So this is method number 1. There are 2 basic methods of forming the mesoderm. Method number 1 is where often just a single or a few endodermal cells, that is, cells separate from the endoderm here and move into the space here, move into the mesenchyme, and as those cells proliferate, they give rise to the mesoderm. So here's the beginnings of the mesoderm. Here's the ectoderm on the outside, the endoderm lining the gut. The blastopore is the opening to that gut, which will eventually become a mouth or anus. And in general, again, this is not a hard and fast rule, as it turns out, but in general, the formation of a mesoderm by splitting off cells and migrating in here is generally associated with organisms that have spiral cleavage. Not always, but usually. All right. So as those cells move in into that space, into the blastosphere, they expand and form the mesoderm. So here we have, again, we're looking at a cross section here. So instead of looking at the opening end, so this gut does open to the outside. This is not shown in this section here. It opens at one end. So here we have the gut. We have the endoderm surrounding it. We have the ectoderm on the outside. The white spot is the blastocele, the original cavity inside the blastula. And then the shaded gray parts here are the mesoderm that's expanding. In animals where the cells basically just split off the endoderm and move into the mesoderm, they expand and eventually they just split. And that split in the mesoderm forms an internal cavity that becomes a true coelom here. So this is a true coelom. Formed by the split. And notice that the space here, the white space that used to be part of the blastocele, eventually gets squeezed out and eliminated by the mesoderm. And these spaces here instead become the internal cavities. And this process is called schizocele. Schizo meaning the split, and cele meaning cavity. You form a cavity by the splitting of the mesoderm. So that's mesoderm and coelom formation. The first type. The second type is, again, we start with the basic gastrulation here. You're forming a primitive gut. And as that gut begins to develop, you have outpocketings from that gut. That is, these evaginations or outpocketings from the gut here. You still have the blastopore opening. And these outpocketings of the gut expand and then separate themselves from the gut so that they become separate. So they basically fuse together here. So the gut is completely lined. And then this tissue here and here forms the mesoderm. And the space inside of that that's trapped in here in the coelomic pouch, which used to be connected to the gut, the archenteron, then becomes the coelom. So again, you have two. So here you're forming the coelom via that's called enterocoely, where the coelom is formed by being basically trapped in these outpocketing guts here. So it's originally was basically an outpocketing of the gut that becomes sealed off from the gut and captures a cavity inside that way, and that's the coelom. The end result looks pretty similar, so it's basically, you can't really look at an animal in cross-section and determine if its coelom is developed, which way the coelom developed. You basically have to kind of actually look at the process of development. But the key thing in both of these cases is that the mesoderm comes from part of the endoderm here, either as separate cells that migrate into the space or as an outpocketing of the gut of the endoderm to form a separate layer. Okay. All right. So the final aspect is, so let me go back to here. So notice here we have an opening. The archenteron is the ancient gut. It has a single opening, the blastopore. Here, this is looking at it a different way, so you're not seeing the opening, but the opening would still exist. And in all of these cases, the gut here has a single opening. But many animals have digestive tracts with two openings. We call that would be a complete digestive system. An incomplete digestive system has a single opening, which is derived from the blastopore, which functions both to take food in, but to also eliminate undigested waste. So that acts as both a mouth and an anus essentially. But animals with a complete digestive system have two separate openings. They have a mouth at one end and an anus at the other. And so that means that food can mostly move through the digestive tract in a single direction. It can allow for much more specialized digestive tract and so on. So what that means is that when you start with a gut that has a single opening, the blastopore, that blastopore can basically develop into the mouth, which means you need to form a second opening, the anus elsewhere, or vice versa, that blastopore can become the anus and the mouth forms secondly. And so that gives us two more kinds of development. Protostomate versus deuterostomate development. If you've heard of any of these terms, it's probably these two in introbiome. So proto first, stoma meaning mouth or opening. So basically in protostomate development, the mouth forms first. In other words, the blastopore, that first opening, becomes a mouth, and the second is the anus is added afterwards. In deuterostomate development, deutero second, so the second mouth, the blastopore becomes the anus, and the mouth forms second in the development of those organisms. All right. And so the reason you've probably heard these terms, protostomate and deuterostomate, is because we also tend to use, for a long time, use this as a basic system, basic way of classifying organisms. We used to think that this was pretty development, this kind of basic development was phylogenetically important, and it still is, but not as important as we used to think it was. And so we still use the name protostomes and deuterostomes for two major lineages of bilaterally symmetric animals, bilaterally symmetric triploblastic animals, that are distinguished typically by whether they have protostomate or deuterostomate development. However, as it turns out, this is not 100% reliable, and some animals, which molecular data tells us grew very clearly with, for example, the protostomes, actually have either some or mostly deuterostome type development. So that's why the typical is kind of emphasized here. Most protostomes have a protostomate kind of development, but there are exceptions to that. But we still use this group to refer to that group of animals taxonomically, even if they sometimes have a deuterostome type development. All right, so as it turns out, that many of these features of development tend to be, again, there's a lot of Weasley words here, so typically tend to show a suite of developmental features in common. So the animals that we recognize as protostomes, typically, again, there are exceptions, things like the lophophorates, brachiopods, bryozoans, which you probably haven't heard of yet, things like arrowworms, which you almost certainly haven't heard of yet. But a few various things, mostly more obscure groups, tend to be exceptions. But technical protostomes tend to have spiral and determinate cleavage. The blastopore forms a mouth, that's what the name protostome means. The mesoderm for animals that have that kind of development tends to split from the endoderm, and that is, a cell separate from the endoderm, and the coelom is formed via schizocoely. These two generally go together. A few other things that occur later in life is that most protostomes, the central nervous system, the main nerve cords of the central nervous system that send signals from the brain to the body, tend to be in the ventral portion of the body. They tend to be on the belly. The larvae, if they have cilia, tend to have compound cilia. That is, the cilia are derived from multiple individual cilia, and there are multiple cilia per cell. The number of coelomic cavities, that is, let me go back to this here. So when you form a coelom, you first of all form a right, there's usually a separate right and left half to the coelom here. But if the animal is segmented or elongated, you can also have this process occur in multiple sections, forming several independently formed sets of cilia along the length of it. So you can have multiple coelomic cavities that are produced by the same mechanism. And so the number of coelomic cavities in protostomes tends to be very variable, from just the two, the right and left, to multiples. Examples of typical protostome animals include arthropods, annelids, segmented worms, mollusks, and so on. The deuterostomes, in contrast, have radial and indeterminate cleavage. The blastopore becomes the anus, because deuterostome means second mouth. So the first opening is the anus, and the second is the mouth. The mesoderm in typical deuterostomes usually forms as an outpocketing of the wall of the archenteron, of the primitive gut. And the coelom therefore forms by enterocoely, by being captured in that outpocketing. Most deuterostomes typically have a dorsal nerve cord. They usually have just single cilia, one per cell, and the cilia are single in the larvae. And usually deuterostomes tend to have 3 pairs of coelomic cavities, at least early in development. Again, that can be changed in adults, but usually during the early development, they start off with 3 pairs of cavities. And the examples you're most familiar with are probably the echinoderms, sea stars, and things like that. And of course, us, the chordates, the vertebrates. We are also deuterostomes. Most of the animals that we tend to think of as deuterostomes have pre-deuterostome type development, so these groups are all pretty consistent. But the protostomes tend to not be quite as consistent. There are a number of animals that phylogenetically show up in amongst the protostomes that have either some or all or most of the aspects of the deuterostome development. So again, this is kind of a nice sort of classic view here, but there are quite a number of exceptions, even if it sort of typically holds true. There are many exceptions, that is. Some protostomes might have a coelom that forms via enterocoely, for example. They might have different types of larval cilia. Those aren't all, none of these things are necessarily 100% consistent. Okay. All right. So I think maybe it's, so yeah, various animals deviate from the general deuterostome pattern. And in fact, in some groups, including some of the lophophorates, these are animals with a sort of a ring-shaped group, group of ciliated tentacles, things you probably didn't cover in intro bio. But some of them, even within the same phylum, even within the same class, you can have animals with somewhat different development, at least some that differ in some of these basic developmental things. Some, like arrowworms, have pretty much a normal deuterostomic development. Molecular studies tells us they're protostomes, despite the deuterostome-like development. It's generally thought that a deuterostome development may have been the ancestral one, and then protostome development evolved several times. In addition, the lack of, so notice that when we go through that one, I'm not going to go back all the way, just flip back to the pictures, but when we form those coela, we form the coela more or less at the same time as a mesoderm. In very small animals, it might not split, but if you're forming your coela via enterocoely, you're going to basically, if you're forming your mesoderm, you're going to have at least some basic coelom there. And then what can happen is that coelom can just simply close up. That is, you can squeeze out all that fluid and become solid. And so some animals that lack coela, most famous being flatworms, the Platypum nymphes, they probably, their closest relatives are maybe things like gastrotrix or rotifers, which tend to be pseudocoelomate. And so that loss, that fact that they're acoelomate probably represents a secondary loss of the coelom. And the same with pseudocoela, which is essentially sort of an embryonic stage where you're retaining the blastocele as part of your coelom. That has evolved, the retention of a pseudocoelom has basically evolved multiple times within the history of animals, and probably is associated with small body size. As you evolve to become smaller, you do that by basically sort of arresting your development at a juvenile stage, so they've retained the sort of ancestral, the sort of juvenile characteristic of having only a pseudocoelom, having that original a remnant of that original blastocele. Okay. All right. So what about beyond your basic early development? Beyond early development, then we can get into the kinds of life cycles that animals exhibit. And again, you can have two different life cycles, direct versus indirect development. So in indirect development, you basically form a larvae that is distinctly different from adults. At some point, that larva that doesn't look often anything like adults will have to undergo a fairly significant metamorphosis, a rearrangement of its body to look like the adult. So indirect development is you have a free living larva that doesn't look like the adult, but is nonetheless free living. Those larvae can be planktotrophic, and again, we're cutting out lots of terms here, but these are kind of the important ones. So a planktotroph, plankto meaning plankton, that means that's the stuff, the small little organisms, both algae and bacteria and animals that are up in the water column off near the surface. Trophic meaning feeding. So planktotrophic are larvae, are larvae that live in the plankton. They're free living up in the water column and feeding on their fellow plankton. They may be feeding on bacteria or algae, or unicellular algae or something like that. So that's a planktotrophic larvae. In contrast to a leucithotrophic larvae, leucitho meaning the yolk. So these are yolk feeding, essentially, is what that name translates to. These are larvae that depend on a large, well-developed yolk for its nutrients. They can still be free living, but they have a large yolk, so they don't have to go feed for themselves, which means they don't necessarily have to get up into the water column. They can remain wherever their eggs were laid, or perhaps even be retained in the parent for a while. But they can depend on that yolk for a lot of its nutrients for the earliest stages. That provides nutrients and energy and so on, and resources to allow them to undergo the development and become larger. One of the things to kind of keep in mind about the evolution of different life cycles is that if you take, let's say you take something like a crab. Crabs are really, really complicated, And if you start off with a single tiny little egg, that egg is not going to be able to develop into a miniature crab right away and be functional at very small sizes. That is, it's generally not practical to take a large, complex, multicellular animal, shrink it down, and still keep it functional. Some parts just are too complex to be able to be shrunk down. You end up losing material, or they just may not be able to interact with their environment in the same way a larger adult would be. And so that's why you have different larvae. Larvae are designed to function. They may just be a flat little disc of cells that's covered in cilia that allow them to swim, while an adult might might have legs and things like that to crawl around the bottom. At a very small size, those legs you wouldn't really be able to operate them. And so you have a very different kind of larvae. And sometimes you can have multiple stages of larvae. If you have larvae, if you have young that you release into the environment at a very small stage, they often have to be, just sort of out of practical necessity, substantially different from adults in order to be able to function at those small body sizes. And remember, you're also going to be relatively simple in those early stages as you're just starting your development. All right. In contrast to indirect development, in direct development, you're producing your eggs eventually are developed into a little miniature-looking adult that has basically all of the features or most of the features of the adult before it's released into the wild. So the embryos, before they can actually take on a functional shape, basically can't be free living, and so have to be either brooded in, such as inside the female, so she's basically pregnant, or contained in some way in the parents, or they can be put into a protected environment, like for example sealed up in an egg case, where they can rely on the yolk to continue their development until they're large enough to actually function with an adult-like shape. And so they emerge. So when they do eventually hatch or emerge from the female or from the egg case, they generally resemble the parents. And as you might imagine, in direct development, you have to produce pretty yolky eggs. You have to give those young enough nutrients that they can complete enough of the development to get large enough to basically be functional in the adult body shape. And direct development is most common in both freshwater invertebrates and terrestrial invertebrates. And if you think about it, lots of larvae are designed to be planktonic, to go up into the water column, to just kind of drift about. They don't even have to swim very much. They drift about, bump into food, find little bits of algae and so on to feed on. So they can be functional in a very simplified body plan. On land, you can't be planktonic. There's nowhere to go. And in fresh water, you again have to be relatively sophisticated to be able to be free on your own, because in fresh water you have osmotic regulation problems. That is, you're at a very different internal concentration of salts and proteins and sugars and things from the environment. So that means you have a natural tendency to take up water, which means you have to osmoregulate fairly actively, which means you need an osmoregulatory organs and things like that. So you have to be reasonably well developed in fresh water before you can kind of face the environment yourself. So you're retained in your parents typically, or in some sort of egg case that can retain the right environment for you. So you don't, until you're large enough to be able to osmoregulate and cope with the difficult environment of being in fresh water, or even the more difficult environment of being on land. And so most freshwater and terrestrial invertebrates, will skip insects for the moment. Insects are a special case, tend to have direct development. Young develop inside of a protective egg or egg case or inside of the female until they're large enough, until they're developed enough to be able to function on their own, looking like miniature adults. Okay. So, when you think about the various kinds of advantages and disadvantages here, these are not just random life cycles, they're basically different adaptations to certain environments. So if you have indirect development of planktotrophic larvae, you're producing generally tiny little larvae that, especially in their earliest stages, might not look like much of an organism at all. They may just be little spheres or little flat discs or worm-like things covered in cilia. Why might this be kind of favored? Well, if your parents are benthic, meaning they're living at the bottom of the ocean, and if they're sessile, meaning that they're attached to the substrate, can't move around, if you can't move, then how do you disperse? How do you invade new habitats, spread into new environments, and so on? Well, you use your larvae to be the disperser. So you need to have, if you're as an adult, relatively immobile, then you want to use your larvae, which can drift around in the plankton until they're big enough to settle out. So having having a larva that's a different appearance of you, a different lifestyle of you, has an advantage, as well as it allows for dispersal. So we find indirect development is associated with broadcast spawning. That makes sense. If you're spawning your eggs and your sperm into the environment, those eggs aren't going to be brooded by parents. They're not going to be in an egg case. That means that as soon as that sperm fertilizes that egg, that developing zygote has to more or less be able to function. I mean, for a little bit of while, it can maybe use a little bit of yolk, but it starts to need to get food on its own. So it often forms a basic form that is able to do that while drifting around in the plankton. So indirect development of planktotrophic larvae is going to be closely associated with species that are broadcast spawners. And broadcast spawning is particularly helpful if you're attached and can't move around to find a mate, for example. And we generally find these indirect development is fairly common. Again, it's mostly in marine environments, not freshwater environments. I want to clarify that. We typically find them in tropical and coastal areas where conditions are reasonably predictable, where you can be reasonably assured that your larva is going to settle out somewhere where it can be tolerated. And ecologically, they tend to be R-selected. You guys know what R-selected means versus K-selected? No? Okay. So it's basically a life history strategy thing. At some point we'll probably cover it. So an R-selected species is one that tends to produce lots of small young, producing in large numbers, but doesn't invest very much at ease. So they tend to have high mortality. And it gets the name R from the population growth rate. They can produce lots and lots of young, so they have the potential to produce high growth rate, but also high mortality, since parents tend not to invest very much in them. All right, compare that to indirect development, again producing larvae, but where your larvae are leucithotrophic, where rather than letting your larvae out into the plankton, your larvae may be mostly getting most of their free living, that is, they're in the environment, but they may be getting most of their nutrients from yolk rather than having to fend for themselves. So indirect development of if you have leucithotrophic larvae, that means that there's more parental investment in the offspring, in the eggs in particular. You're producing bigger eggs. That means the female has to yolk up those eggs, put more nutrients into each individual egg, which generally means if you have to produce more of something, you only have a fixed amount of resources that an animal has available to it. So if you produce bigger offspring, that generally means you're going to produce fewer young. But because the young don't have to survive on their own as much, they can basically be somewhere kind of protected and therefore get a head start by relying mostly on their yolk. So that generally means they have higher survivorship. So that's the trade-off here. You produce few young, but because you invest more in them, more of them are likely to survive than if you produce planktotrophic young, where most of them will die. Leucithotrophic larvae tend to be found particularly prevalent in deep sea species. Various invertebrates like sea lilies and brittle stars and other weird things, sponges and so on that live in the deep sea. This kind of strategy is more common there. The reason is that if you're in a deep sea, producing planktonic larvae, where am I on time? Producing planktonic larvae basically is even riskier than if you're in a nice shallow coral reef or something like that, because it's much further to go from where you are to the surface. So there's a greater opportunity that someone's going to intercept your larvae and eat them. And then they're going to have to grow up in a very different environment in the surface waters before they try to settle down, possibly thousands of meters of depth. So that risk to the surface and back is pretty high for the larvae. So you're better off investing more, keeping them in the deep where there isn't much food, and so they have to rely on the yolky eggs for their much of their early development. Okay. And then the final bit of life cycle is direct development. Again, in direct development, you're relying on large, costly eggs. There may be additional parental care as well if you retain those eggs in your body, for example, or in other ways provide for care. Direct development is usually associated with mobile adults that can move around on their own. So that means you don't need the young to disperse because you're the dispersing stage. And so you can keep the larvae in either with you or in some protected place. You can bury them, put them in a burrow or something like that, attach them to the substrate somewhere where they might be safe. So typically these are sort of ecologically more K-selected in comparison. The letter K refers to the symbol used for the carrying capacity of the environment. So ecologically what that means is these are species that produce relatively few young, invest very heavily in each, and so the population tends to be more stable, at carrying capacity, they have less potential for increasing. They don't increase as fast because they're not producing as many young at any time. But it tends to be more stable because you're investing a lot in each young, so there's a good chance that a significant number of those will survive. Direct development is also typical of terrestrial species, where things like gametes, broadcast spawning of your gametes is not possible in terrestrial species, and it's really not practical in fresh water either because of the osmotic differences. And so in freshwater and terrestrial species, those really delicate little planktotrophic larvae are just not possible at all. And in fact, because you need to kind of keep the eggs inside the female in most terrestrial species, you also need internal fertilization. So that's another feature that occurs in many marine animals, but also is almost pretty much a necessity for terrestrial species. So trying to think of any exception. There may be some exceptions among freshwater species, but certainly I can't think of any terrestrial examples that don't have internal fertilization. All right, so with that, we're going to stop. There is a special case. So notice I said that most terrestrial species have direct development. That's not technically true. The most abundant terrestrial species are insects, and many insects, so those that are particularly abundant, actually have secondarily evolved indirect development. So we'll deal with insects, a special case of insects, next. But in insects, you still produce large, costly eggs. You are producing young that are basically terrestrial as opposed to simple, delicate little larvae. They're just different